katoa. Welcome to the third of our Mammal Pest Talks for Biosecurity Bonanza 2024. It's my pleasure today to introduce Dr. Patrick Garvey, a wildlife ecologist at Manaaki Whenua. Patrick's work specialises in the behaviour of invasive mammals and how we can use their behaviours to improve control. His talk today will take us through the findings from the Eradication Science Research Programme. A Q&A session will follow this talk. I encourage you to submit questions in the chat during the presentation. We will try and get through them all, but if not, Patrick will respond by email to any questions we don't get to today. The session is being recorded and all recordings will be available on our website within the next week. If you'd like to connect with the presenter on project ideas or future opportunities, you can make contact via our website and then search their name. I'll now hand over to Patrick to share his presentation, Don't Be Shy, Understanding and Targeting the Survivors of Pest Control Operations.
Thanks very much, Sam. Good morning, everybody. Thanks for joining. Today, I'd like to talk to you about our Eradication Science Programme that's in its final year of a 5-year programme. This programme's co-led by Graham Hickling and it involved people across the land care team. So today, I suppose I won't be going into too much details. It'll give an overview of some of the research we've done, but I'm happy to answer any questions in more detail if you've anything you want to ask. But basically, it'll be a whirlwind view of some of the more exciting research that we've had so far. So undoubtedly, invasive predators have dramatic effects on biodiversity, and I'm preaching to the choir here because this is very well understood by this audience. And we also know that invasive predator control is one of the most effective conservation interventions. It's highly effective, and when compared in a recent paper by Langhammer in Science paper, he looked at different methods and the effect size of invasive predator control was the greatest. So it is the most effective intervention method. So we know invasive predators are a nightmare for native biodiversity. We know that invasive predator control is effective, but one of the challenges we have is those animals that won't engage with control tools or devices, and they are the survivors of the control operations. So when we think about survivors, what are the issues with them? First of all, they facilitate recovery. So the more survivors you leave behind, the quicker the population recovers. They maintain predation pressure on biodiversity. They're difficult to remove. That is one of the great challenges for the predator-free initiative. And they're expensive to remove. And what we see is that the cost of removing a pest at the outset of an operation or even into the middle towards the end is relatively stable, but as we get to those final few survivors, the ones that refuse to engage with devices, the cost increase exponentially. So survivors are a real challenge. So today, just to give you an overview of what I'd like to talk about, first of all, I'll talk about individual variation, survivors versus the average pest. How are they different? We look at behavioral responses towards traps and baits, and there'll be a particular focus on personality, animals' perception of risk, and how they respond to control tools. Then I'll just chat about some novel lures. These are based on a framework called the 4F motivations, which I'll explain. We're looking at sensory cues, both sound, scent, and visual. And the challenge is whether these lures can overcome survivors' behavior. And finally, I'll talk about targeting survivors, collaboration. We are currently in collaborations with control programs across the country and our HAPU partners. And we use the 4F lures that I'll talk about in combination to try to target survivors. And the field trials of the most promising lures are currently underway. So first of all, just understanding survivor behavior. So have survivors any particular characteristics that make them more likely to survive? And the sort of things we were looking at was a particular focus on animal personalities, but we didn't just base it on personalities. We also looked at intrinsic differences, such as the sex or weight of the animal. We looked at whether avoidance or survival is linked to a learned or conditioned behavior. We investigated diet and microbiome, particularly microbiome, not diet to a great extent, but that's another important factor. And our Other hypothesis is that survivors are just a random subset of the population. It's just you remove some, there has to be some left, and there's nothing, it's a density effect rather than any characteristic of the survivors themselves. So if we think of a pest population, I suppose traditionally pests are managed as if they're a homogeneous group. So all individuals are essentially the same. If we put a trap down, one would enter and put something like peanut butter for these rats, one would enter, then we reset, the next one would enter, and you keep going until all members of the population are gone. But what we know from research in animal personality fields is that there is different characteristics within this population. That rat might be shy, the other bold. One might be neophobic, that's scared of or not attracted to novelty, and one neophilic, which means attracted to novelty. One might be inactive, stays relatively close to home, where one might be venturing further afield. So if we think about these characteristics that's in the pest population, we have to wonder of whether these uh, characteristics will influence their responses to bait or traps, or could this be one of the underlying reasons for survival? So when we think of animal personalities, personalities are defined of behavioral differences between individuals that are consistent through time and across context. And the big five personality measures that are used in animals are boldness, exploration, activity, aggression, and sociability. And the three we look mostly at are the first three: boldness, exploration, and activity. And these link across to the fitness of the animal. So these traits relate to the survival and reproduction rates of individuals, their home range and how their range expands when neighbors are removed. The dispersal distances are linked to boldness, activity, and exploration. And the diet of the animal can be linked to these traits. And also the risk taking, which, as you would be obvious, it's an important characteristic that might be important in the context of wildlife management. So these are the traits of the animals that we look at, and this is how we measure those traits. So this first arena, you can see a stoat wandering around a darkened area, is called the hole in the wall. You release an animal in here for 5 minutes, and you look at how they move through the 4 segments that are equal size. The 2 at the back are lit up, so these are more dangerous, perceived as more dangerous to the animal, and it's how the stoat will make use of those space areas. And what we do is we measure it once and then measure again 2 to 3 months later, then measure them again 2 to 3 months later, and repeat the test 3 to 4 times, and then try to identify consistencies in behavior throughout the test related to these trait measures. Another approach we use is a novel object test. This is where an animal is in its nest box, it comes out, and then it encounters an object that it's never seen before in its life, such as the slinky or toy car. And then we look at how long it takes them to approach and interact with the device. And then by repeating the test, we can assess how they respond to novelty in comparison to other members of the population. And finally, the startle test, which is a pretty cool test, is we put a camera over the nest box in the cage. We wait for the stoat to be out, and then somebody, Kyla in our case, walk down the aisle just muttering random words, and then you look, the stoat obviously runs away when it sees the human, and the measure is how quickly it re-emerges in the face of this risk, and then that's repeated through time as well. So these are the types of personality measures we use, and now we can see an actual experiment where we try to link personalities to avoidance or interaction with devices. So this trial was undertaken at the animal facility. We tested 3 stoats at a time with a separate cage in between each of them. And we looked at their interaction with 3 types of traps. One was the box trap, which is a dock and it's open at both ends. One was a tunnel trap sealed at one end that's used for stoats and weasels, and we had an unset Victor trap. And finally, we looked at a head-up device like the A24 in the picture, and looked how stoats interacted with these devices. In total, we had 28 stoats that were measured. So this just shows the interaction rates with devices without incorporating personality. And you could see the dock box trap is highly effective. 75%, more than 75% of animals interacted on the first approach. we had the trap out for 15 hours, an opportunity for each animal to interact. So you can see on a subsequent encounter in the grey area, although some animals didn't enter immediately, some subsequently entered, and then there is one animal at the top of the box or dock trap that failed to interact, and I'll talk about her a bit later. And if we looked at the head-up device, it was comparatively very unsuccessful. Less than 10% interacted straight up, and only about 25% overall. And then with the tunnel trap, we had around half interacted at the start, another quarter subsequently, and a quarter never. So you can see the different device types invoke different responses from the Stoats. But then if we incorporate personality, how can we understand the individuals that are avoiding the devices? So the novel object test, which I'd previously highlighted, if you looked on the y-axis there, you can see the time to trigger the device, and at the bottom is the time to contact, which is a measure of boldness. And you can see that the animals that were very quick to contact a novel object or unusual object they hadn't encountered before were also very quick to trigger the device, and there was a significant relationship between these two factors. So the animals that avoided the device were also the same animals that avoided the novel objects. And similarly, when we look at the startle test, another measure of personality at the bottom, we can see the latency to re-emerge after a human walked down the aisle was directly linked to the amount of time it took to trigger the device. The animals that reappeared quickly after a person walked by were also the animals in a completely separate trial that would trigger the device fast. So this shows the link between personality and device interactions. And at the predator-free HUI, I was asked about rats, so I included some information on rats that I hadn't included before. And again, similar trial setup. In this experiment, what we did was we just had a non-toxic bait on the ground, and that was basically our control. There's no device. We looked at the amount of rats. We had 46 rats that were tested, 26 from sites that were controlled and 20 from uncontrolled sites. And we looked at the number of animals that would eat the bait on the ground over a 15-hour trial night, compared that to interact with or to remove a bait from a dock trap, compare that to remove a bait from a head-in trap, and compare that to remove a bait from a bait station. So these are the 4 devices we looked at. And here we can see that the bait on the ground, unsurprisingly, was interacted by most animals, but indeed not all. About 90% of animals took the bait from the ground over the trial night. In comparison, from the dock box, it's about 75%, generally high interaction rate. The head-in device is much lower again. 25% were willing to remove the bait from that device over a trial night. And the enclosed device, the bait station, had the least interaction in our experiments with these 46 individuals. And then if we compare that to the personality of the animals, we had a measure of boldness, which is the release test, how quickly an animal goes back into its nest box after being released. And you can look at each of the treatment types. For the ground, there was the least interaction with the trait that relates to boldness, whereas for the enclosed device, it was the highest rate, it had the highest effect. So basically what we're seeing is if the personality, the most risk-averse individuals are least likely to interact with a control device, and that is related to their personality, head-in then second, and a dock and the ground much more likely to interact. So can we see evidence from the field? One of the questions you'd say is, okay, you can see this happening in a pen, but is this real? How real is it? Will we see examples if we look to the wild? So we did a bait station operation over around 6 to 8 weeks at Joseph Langer Reserve, and you can see the blue line represents an untrapped population that was an area where it hasn't been controlled, and the red line represents a trapped population. And at the bottom on the x-axis, we look at time to interact, a measure of neophobia and neophilia respond to novelty. So what we see is that you're missing the bold animals. The bold individuals are completely absent because they've already been killed. When we put the bait stations out there, it's the animals that are quickest to approach novel objects, are quickest to investigate novelty, that are also the ones that take the bait from the bait station. So you can see it's completely truncated. We're missing all the bold animals from an actual operation. And similarly, and this is a study that was led by Graham Hickling and Lottery Bush, but we caught possums before an operation, and on the left-hand side you can see the spread of traits from the hive being the shy animals and the lower being the boldest animals, so there's traits were spread across. When we caught at the midpoint, only half the amount of animals. We caught 20 animals again, but their personalities had shifted much more towards the shy end of the scale. And by the end of the operation, the animals we caught were extremely shy, again highlighting that control operations with traps just remove the boldest individuals. So that's a kind of understanding of where we stand with traps. But what about baiting operations? Is it similar where the personality of the animals influences who survive? So this was an operation that took place at Joseph Langer. It was a single control operation followed by a single pre-feed. There were RS5 baits that were 6 gram. And what we did was a crazy experiment where we monitored the animals through the operation to see who would, or is there any characteristics that were linked to survival. So how this looked, we captured 128 possums in total. 24 of them were collared at the site and released prior to the operation at the site, but we hadn't brought the captivity. Then there was some juveniles that we just released back automatically after ear tagging. 85 were brought back to the facility, brought across the country from Franz Josef to Lincoln. There we measured their personalities in book tests, and we also did a feeding trial of a non-toxic version of the bait to see their acceptance rate. We returned these 85 animals to the site. At the point of capture, each was released again with a collar, and then the operation happened. So then the 1080 drop occurred. And at the end, after the operation, from the 128 individuals, we only had 8 survivors, of which we recaptured 7. We had 8 new survivors that hadn't been captured previously that we also captured after the operation. And we sped these back to the animal facility to see how these different from the ones that died. So just in summary of the results from the baiting operation, as I've spoken about this previously, but this will give an overview. Baits were not perceived as scary. From the 85 animals that we brought back to captivity, all 85 were willing to eat a lethal dose of the bait before the operation. But after the operation, these same survivors refused to eat them. So sublethal dosing or sublethal poisoning or conditioned taste aversion was a key mechanism that linked to survivorship. And bait avoidance was sustained through time. When we tested the same animals nearly a year later, most continued to avoid the bait. And there was a clear trend towards juvenile males. These were one kind of increased numbers in survivors and also large individuals, both which can be explained. Larger individuals because the smaller bait were more likely to lead to sublethal poisoning. But there is some good news with this. All 85 possums, as highlighted, were willing to eat the bait. There's no evidence that survivors could detect 1080 because when we switched to different bait matrix, we killed nearly all survivors. Encounter was not an issue with a single bait application. All 128 animals in the operations encountered baits. And bait switching killed all but one survivor. So there was still one champion that made it all the way to the end. Out of these 128 animals, there was one animal that refused to eat, that survived the operation, refused RS5 and refused the carrot. But we can really, if we get better at ensuring an animal gets a complete bait, we can really drastically reduce the number of survivors for an operation. So just in summary on survivors, trap survivors are extremely shy, they're less active, and they're neophobic. Traps and bait stations are perceived as dangerous by these individuals. Control operations leave a remnant population of shy individuals that we can see from operations in the field, and female stoats tend to be more risk averse. From the 5 animals that avoided devices in our pen trial, they were all females. And then with bait survivors, activity is linked, so less active, less exploratory animals were more likely to be sublethally dosed, less dominant animals, but this is linked more to how long it takes them to encounter baits and bait degradation. Baits were not perceived as dangerous, they were all willing to eat baits. There's a trend towards juvenile males and large possums, and survivors, the issue was the survivors failed to encounter a single lethal pellet when they first encountered a pellet. So fast acting toxins ended up leading to condition taste aversion. So that's kind of our understanding of survivors. But we understand them, but that's only half the problem. We need to be better at targeting. So this is the focus of the second half of the study, targeting survivors. And if we imagine there's a rat in the forest and it's just running around eating everything it encounters, murdering everything, and all of a sudden it encounters a trap. Now, whether that rat enters the trap is based on a few different factors. First, the individual characteristics of the animals, like we highlighted with personality. Secondly, the perceived danger of the device. Thirdly, the background environment. For example, how hungry is it? That could lead it to interact where it would not necessarily do so under normal events. And also the sensory cue, how attractive is the lure in the device? So all these factors kind of interact together, but the individual variability we can't do much about, we can just understand. But I'll quickly touch on the background environment and perceived danger, and then focus on the sensory cue. So what we're seeing from the background environment is that it can influence whether an animal is trapped in law or not based on what's happening in the background. So, for example, we do our aerial baiting when food resources are low in winter because the background environment has low food and it makes an animal more likely to engage with the baits. Similarly, we could bait match based on resources, seasonal resources, or we could use things like pheromone lures during the breeding season that will target, because the background environment, the animals need to find mates. So this is kind of ways we can consider the background environment when targeting pests. And then we have the perceived risks of devices. And what we really need to understand is that pests are under selection pressure to recognize and avoid devices. So in order to overcome the challenges of the most difficult survivors, there are a few methods that could be used. Pre-feeding is something that's standardly done to make sure the animals engage and create scent trails. High value baits increase the value of reward. Passive devices, very important, especially for those animals that refuse to engage with devices at the end, the survivors. If we had more natural traps, more natural looking traps, these survivors might be more willing to engage. And low risk traps, I highlight the Victor trap there because it's over 100 years old, but it's still being used because the animal doesn't have to enter into any enclosed space. It's a flat surface, so it's more likely to interact with these devices, and that's why a device like that is successful. And just to mention experiments we've completed under the Eradication Science Program, the results are still coming in. We've looked at the same trap. If you alter the material from wood to metal to plastic, the exact same trap, how does that influence interaction rates? We've looked at trap switching in live operation and whether camouflaging traps can increase survivor interactions. And we've also looked at high interaction traps. We've collaborated with Cacophony and looked at how their device, whether it's better at targeting those remnant individuals of a population. So there's studies that we've completed, but results yet to come in. And finally, I just want to talk about this framework that we've used to try to identify new lures. And the framework is called the 4Fs of animal motivations. So all animals have primary motivations, and we're kind of targeting those drivers to see can we identify new types of lures. So the first F is feeding, and this is obviously an animal needs to locate food, and this is our typical approach to control. We use baits such as rabbit meat or peanut butter. The second F is fighting. This is where an animal has to defend its territories or challenge other conspecifics for mates, and that could be another area we try to target with new lures. It's not an area that's targeted at the moment. Similarly, fleeing, an animal must survive, so it must be aware of the predators and where they're around. So we could use things like predator odor or predator visual lures to help target these animals. And the last F, I'll leave this for a question. You can tell from the photograph there. That's right. It's fornication. So this is finding mates. So animals must find mates, and such as Stoat kit calls can be attractive to males, or estrus females can be attractive to males, and this is other areas we could target to identify new lures. So when we think about these new lures, the way we did it for eradication science, we tested between 10 and 15 sound, scent and visual lures on each species, particularly focus on what interests the survivors, the animals that refused to engage previously. And the type of lures we use, just to give some examples, we have sounds such as fighting possum, rat contact call, or a baby stoat, and these are targeting different aspects of the 4Fs. We have scent from male and female specific, where they'd have to defend fighting to target that fighting element. We've things like cat odor and mouse odor, or predator odor, prey odor, and we also have visual lures such as glow lures, not glow sticks, fake mice, fake eggs, and stuff like conspecific tail. Can we represent a conspecific, another member of the same species, and use this as an attractive lure? So that's just some examples of what we tested in the pen trials, and now we're at the stage where we're trying to test the best of those in field trials. But just before this, I just want to highlight how we have whether these types of lures can help target survivor. The female stoat, the extreme female, the only one that of the 28 that refused to engage with the dock trap, you can see where she sat on in terms of personality. So on the left, on the y-axis, you have the time to approach novel object. On the x-axis, you have the time to emerge after a startle, and she's right as the most extreme individual. So these are the type of animals that refuse to engage with devices. We tested her for 15 hours on a trial night. She refused to enter with rabbit meat as a bait. Again, tested her a week later. We then tested her 6 months later, again with the same dock trap and rabbit meat. She refused again to engage. So what we did as a last resort, we used predator odor as a social odor, ferret odor, and placed that in the trap with the rabbit meat to see if this might overcome her reluctance to enter. And when we tested that, it only took 8 minutes for the female to enter into the trap. So I'm not suggesting that this is a panacea, but it is the type of odors that we can use and type of cues that we can use that can overcome avoidance in some, at least in some individuals. And just quickly to highlight the field trials that are underway, and also to say thanks a million to all our collaborators out there that are helping us to test these lures. On Secretary Island, you can see a map of the current distribution of predator odor that's been tested on the island, along with standard rabbit meat. Five Finger Peninsula, we're working with Pure Salt and Rusty there, and one of the cues he's testing is cues that we identified in pen trials. We're working in Living Spring, testing scent, sound and visual lures, trying to target possums there. Summit Road Group, there we're looking at bait stations and whether bait stations can be lured similarly to devices, to a trap, because if animals respond, maybe they can be lured as such. And there's also some student research underway. Jess Wagner in Victoria is testing sound and scent lures to attract rats, and Laura Grant has looked at social odors and increasing interaction rates with traps. So a lot of studies underway, a lot of people to thank for contributing to this, but I suppose the real utility of our research will only become known in the coming few months and maybe year. And we're hoping to identify a few, 3 or 4 really effective lures that can target these survivors. So just in conclusion, baits are highly palatable and not scary. The fast-acting 10-H toxin leads to survivors and conditioned taste aversion. Baits avoid and sustain through time, and bait switching will target nearly all individuals. When we consider traps, traps select for certain personality types. They leave certain types of individuals. Survivors are shy, neophobic, and less active. And to target the survivors, we can use a range of different devices, attractive lures, passive traps, and one I haven't highlighted but highly important is dogs, to target those very last individuals that refuse to engage. And finally, just the 4F lures can overcome some of these survivors' behaviors. We have a range of sensory lures using audio, visual, and olfactory. And field trials are currently underway. We're really hoping we get some nice results, and we're going to share them with the group and provide any lures and what was the best lures on our Eradication Science website at the end of the trial. So just highlight some of the people we've collaborated and the happy we've collaborated with as part of the project. Just want to say thanks very much to everybody, but particularly for Manaki Whenua staff that have done a lot of this research, implementing of this research. And thank you for listening.
Hi, Patrick. Thanks for a really comprehensive overview of a lot of very technical research. We've got quite a few questions, although I am aware it's 11 o'clock, so we might go through some of them and stay online if you're interested in the results or in Patrick's responses. So the first one is, was our possum survivor a juvenile male?
Yes, it was a male. He's still in captivity. It was actually a juvenile male, yes. So what we noticed, we also did a trial after control operations in St. Arno, and something like 12 of 15 survivors were juvenile males. So we see this propensity to have bait-shy males after an operation. What we don't know is whether that happens because they're slow to get to the baits, being the lowest in the run and the least dominant, so that's why they get sublethally poisoned, or whether these animals enter into the juvenile males are greater dispersers, so whether they enter in after a control operation and quickly appear and get half baits, and that leads to survival. But yep, it was a juvenile male.
And that leads into a similar question. Was there any evidence that the juvenile males had less ability to eat a whole bait, or were they chased away by more dominant animals before they could get a lethal dose?
Yeah, no issues eating bait because we caught juvenile animals and brought them into captivity. They were willing, most of them were willing to eat 100 grams of bait, and a lethal dose is about 12 grams or less. So, and especially for a juvenile, so no issues with eating bait. But for some reason, they didn't get to the bait as quickly, although they were willing to kill themselves, so to speak. They just didn't get a lethal dose of a bait on the first encounter. So they somehow got sublethally dosed partial baits, or because other animals had ate them and left partial baits behind, and that seemed to contribute to survivorship.
Oh, so do you know if the surviving rats or possums spent more time up trees instead of on the ground? And would this account for their behavioural interactions as you have done everything on the ground?
Good question. It is going to be different if a rat is living in a tree and comes down to a bait station that's on a tree compared to a bait station or a trap on the ground. So that is an important factor that we couldn't account for really in pen trials. Also, some of our rats had survived a bait station operation, so that could be also part of the issue. But generally, we just saw that rats were kind of reluctant, being so neophobic, to remove baits from bait stations, at least in our trial when tested on the ground. And it was just interesting to see that bait stations essentially function like a trap, like a head-in device. So although the baits are highly acceptable, once you put them in the bait station, that might be reduced.
There's a question here, how are these traits passed on? Is a shy animal more likely to produce shy offspring?
Ooh, another very good question. They believe that around 50% of personality is heritable. So yes, two shy parents are more likely to produce a shy offspring, but it's not 100%. So nature has a great way of making sure there is some bold and shy within a population, and that ensures bold animals are very important when there's not a lot of predators around and you want to, or where you want to um, uh, move into new areas. So the bolder animals are important in this instance. But yeah, definitely the shy animals have shy traits.
Have you got any preliminary results you can share with us from the study that compared materials, so plastic versus wood, etc.?
Yeah, that's Laura Grant's study. I think I'd be preempting it, but I think the materials didn't make a huge amount of difference. So what we tested was a kind of bait station with a metal wood and plastic. I think the new plastic got least interactions and wood got the highest interactions, but as you know, rats are kind of neophobic anyway, so the interaction rates weren't very high. So what we've done is we've replicated on testing 20 stoats as well to see if they perceive the different device types as different. And they were animals of known personality. So results have still to come in, so not sure, but it seems that the trend was towards wood rather than metal or plastic, but it wasn't hugely so. There's not a highly significant difference, at least in the few videos I watched.
And why would a prey animal be attracted to a trap that smells like one of its predators?
Good question as well. So the jury's still out on that. There isn't a final answer, but what is clear is scent of a predator says that an area was dangerous, not that an area is dangerous. It's not like visual cue or auditory where you know the animal is there. So the sense is free information that's available to prey that they can help them avoid an area or figure out what the predator is and figure out how long ago it was there. So all these factors are embedded in the scent of a predator. So all we know for sure is it's important for the animal's fitness to find out more about the environment. It increases its chance of survival, and we believe that's the reason that they're attracted to the scent of a predator.
Right. And have you got data for different types of trap interactions with rats and possums as well as the stoats?
Yes, and a huge amount for possums. We have over 200 possums interacting with different device types, and it was around 50 rats interacting with device types. But yeah, there's still a lot more data to review. So we stoats were good because they're quite clear personalities and clear behavioral differences that you can see. Rats are more neophobic, didn't interact as much with the devices, but that might change over time if they're left longer with devices. And possums were quite easy. The sample's quite easy to trap, but they definitely distinguish between a device like a Tim's trap compared to a head-in device like a Trapinator. So there was differences, and that information will come out soon.
And following on from that, I've got a question about what you consider constitutes a passive trap.
I suppose, yeah, a passive trap would be a true passive trap would something like something that falls on top of them or goes around them or traps underneath. But I consider a passive trap an invisible trap, one that an animal hasn't to make a decision to interact with it. There's a lure there, but that's all they see, the lure. So generally, I think a passive trap is kind of like a leg hold device, rather than something a cage trap or a dock trap or something like that, where it's clearly the animal can clearly see a device. I suppose that's the difference in my head anyway. One is visible and one is invisible.
So, did you detect any differences in behavior between sexes or different reproductive states of the different species?
Absolutely, with stoats, we had female stoats, the 5 individuals the one stoat that refused to engage with any traps, and the 4 stoats that only engaged with one trap were all female stoats. There definitely was a trend towards, and they had the shyest personality, and we see these in animals that are sexual dimorphic, where the male is much bigger than the female. The females are the other way around. The female is much bigger than the male. The smaller in the sex tends to be shyer, and that's what we're seeing. That's what we're seeing now. That with stoats, the smaller sex is shyer, less likely to take risks. It's clearly evident in stoats. We haven't comprehensively reviewed the data for rats and possums yet, so it'll be interesting to see what's there.
So, how often do the odor cues need to be replaced?
Um, Generally, obviously, you want as fresh as possible. Like when you have a bait, you want it to be fresh, and people notice that when they rebait, they're likely to get a capture. So the fresher, the better. But the great thing about some of these lures is they're naturally long life. Predator odor, like ferret odor, you can smell it 6 months, a year later, after collecting it, you can still clearly smell it on the towel. So some of these natural odors that have developed as pheromones um, are long life naturally, which is quite helpful. But generally, we kind of replenish once a month just to make sure it's fresh. But what would be the ideal length of time is it could be even longer than that.
Cool. And I think this is the last question we haven't answered, but is there a low rate of 1080, sowing rate of 1080 pallets per hectare that becomes counterproductive?
Yeah, I imagine there'd be a certain rate where not all animals will encounter a bait. The great thing about the possum operation is you could see there was different reasons that some animals survive. We had one female that was very shy, very little low movement and activity, and she ate 100 grams of bait before the operation. We recaptured her after the operation, brought her into captivity, and she was the only animal that was more than willing to eat 100 grams after the operation. So what that says is she'd never encountered a bait during the operation, and that might be because she remained in the trees and never came down to the ground, or there was just not a bait directly under the tree she was in. But you do see that the sowing rate can be influential for some individuals, but it, ideally you want to minimize the amount of toxin you're putting into the environment. And it was interesting to see, besides that one female, that every other possum either died or was sublethally poisoned during the operation with essentially a pretty low sowing rate.
Oh, I think we'll leave it there for today. Thanks to everyone who's joined, and the remaining questions will get answered via email. So we'll see you tomorrow. Mā te wā.
Thanks very much, everyone. Thanks, Tam.